So I think recently there's been a number of developments which are exciting. Obviously the increasing capacity of something like the Illumina NovaSeq is very exciting. But what we really need for whole genome sequencing in order to be able to apply that in a clinical context, which is what I'm interested in, is as an increase in throughput and a decrease in price. And so it's encouraging that something like the BGI-seq is coming along, which might perhaps offer an alternative and maybe reduce prices or at least introduce some competition into the market. And then of course there's the possibilities of long-read sequencing, which has a number of great possibilities for structural variant detection. So I think there are some very exciting developments in the last year.
I think one of the most important developments that we're seeing now in whole genome sequencing is the development of long-read sequencing and its ability to be used for analysis of whole genomes. Traditionally, we've been limited to the traditional 2x150 paired end sequencing type of a model where it's very good at calling point mutations and very localized phenomena, but not quite as good at identifying deletions and translocations that really are the underpinning of a lot of the use cases for whole genome sequencing. So I think long-read sequencing is really going to change our ability to do that.
Well, I guess the most important aspect is the fact that there are multiple technologies being used and multi-omics possibilities which are going to reshape the future of genomics.
So in my lab, which is a clinical genomics lab, and we're trying to take some of the latest and most interesting methodologies and move them across into the clinic. And in order to do that, the key driving factor for us is high throughput. It's high throughput and quality. So we need to be able to take hundreds of samples and put those across the same assays at the same time. In order to do that, the logistics of that mean automation, lots of automation, multiple indexing, so that we can take the biggest benefit, if you like, from the technologies that are out there, but also quality control. So all of this stuff has to be done within the context, within the envelope of a very high quality workflow. So all of those things coming together, I think, are going to enable a better clinical application of the sequencing technologies.
I think one of the major problems that we're going to come up against in the coming years as we try to use some of these genomic technologies in clinical use is the ability to manage the volumes of data and the amount of computing power that we need to manage and to analyze the large volumes of data. And I think that's really going to necessitate a move towards the implementation of smaller focused panels of deep sequencing data, moving away from whole genome sequencing types of studies of large cohorts. Once we've learned what the key mutations are, particularly in diseases like cancer, where there are going to be a limited subset of mutations that are actually driving those diseases, I think that once we know that and we can move toward a focused panel, the implications of the amount of data and the ability to handle those data in a rapid amount of time will be paradigm shifting.
Well, the idea is that it will be possible, really, as we have been seen by many presentations today, to bring precision medicine to the individual patient and to be able to understand what are the needs of a specific person, and in relative to its own cohort of very few individuals which share the same needs.
I think high throughput sequencing, so we know that Genomics England and NHS England now are going very big on whole genome sequencing for clinical diagnostics. I still personally think that's a couple of years away for proper application. So the high throughput sequencing methodologies will also be complemented by some other methodologies like droplet digital PCR and perhaps some of the long read technologies for looking for gene fusions, like the nanopore technology, which I think has got great potential for gene fusion detection.
Whole genome and targeted long-read sequencing is going to be a major disruptor. I think the ability to do direct RNA sequencing with some of these technologies is going to change things quite a bit compared to what we can do now. It's going to give us the ability, particularly when married with traditional shorter read sequencing technologies, it's going to give us the ability to have insights on the structural variation that underlies a lot of disease-associated genomes that we just would not have had before. So I think that those types of technologies have the potential to really be disruptive.
I think the future is going to be about how to integrate the data, how to fast and effectively understand the information that comes from multiple sources, integrate existing big sources of omics information together with, let's say, experimental data sets which are built by means of experimental work or clinical studies.
Well, I hope it's going to be reduction in costs and reduction in prices. I think that's probably what everybody else would also want. There are multiple other technologies out there, but of course we're all well aware that there's one kind of big beast provider, and they do have somewhat of a domination on the market. I'm hoping that there'll be new advantages that will allow us to reduce those costs. In terms of new technologies that are coming along, I think the maturity of the long-read technologies is going to offer us things that we're not able to do currently, and I think that's very exciting too.
So I hope that the future of NGS in 2019 and beyond is really a move away from the discovery phase into the translation phase. I think that we've learned an enormous amount about the way genomes are structured, the mutations that underlie risk of disease, underlie pathogenicity of disease, underlie outcomes for disease. And we've really now got to move toward, and the onus is really on us, to move toward a model whereby we're starting to implement some of these changes and really look for tests that implement whole genome sequencing as well as some other targeted sequencing approaches to really drive clinical implementation and make it matter for people.
Well, I'm not going to answer you about 2019. I'm going to answer you about 2025, which is anyway coming up very soon. So by that time, the data which will be produced by Genomics will go beyond, for several orders of magnitude, the data which are produced by other sources, including astronomy, which is currently producing the biggest amount of data, including YouTube, which is also a very big source. So you can imagine the need of people that would be able to process this data, and also the need for technologies which can process big data. These I think to be the area which is most promising, and also the area which I'm going to be involved in the next few years.